Have you ever wondered how pharmaceutical companies can detect just one tiny impurity hidden among millions of molecules? How do we confirm a tablet is exactly 500 milligrams? Not 490 or 510, but precisely within specification? Through rigorous, advanced laboratory analysis. The answer lies in one of the world's most powerful analytical instruments, high-performance liquid chromatography, or simply HPLC. Today, you'll learn what HPLC is. It separates components in a mixture. This is its working principle. It guarantees pharmaceutical quality. Stay with me until the end of this complete series, because I'll also reveal the most common mistakes beginners make while operating an HPLC system. Every tablet, every capsule, every injection, every syrup, every antibiotic must pass strict quality testing before reaching patients. HPLC is the most trusted. It provides exceptional accuracy, precision, sensitivity, and reliability. Whether you're testing the assay of a drug product, related substances, impurities, dissolution stability samples, cleaning validation samples, or finished products, HPLC is used But here's the interesting question. How can a liquid instrument separate compounds mixed together in one solution? Let's discover the science. What is HPLC? HPLC stands for High Performance Liquid Chromatography. Let's understand each word separately. Internal HC means the system operates under very high pressure, allowing rapid and efficient separation. Liquid means the sample travels, dissolved in a liquid called the mobile phase. Chromatography is a separation technique. Different compounds move at different speeds and become separated. This is HPLC in action. Look at these athletes. They push their limits. Every stride counts. It is pure. They all start together. But some are faster. Some are slower. Exactly beside an HPLC column. All compounds enter the column together, but because each compound interacts differently with the stationary phase, they travel at different speeds. Each compound reaches the detector at a different time. This time is called the retention time. Now let's understand the heart of HPLC, the principle. The principle of HPLC is based on the differential distribution of compounds between two phases. First, the mobile phase. Second, the stationary phase. First, the mobile phase. It carries the sample through the HPLC system. Examples include water, methanol, or mixtures. Second, the stationary phase. It's packed inside. Particles are chemically bonded. Every molecule interacts differently. Some compounds have a stronger attraction. They move more slowly. Others prefer the mobile phase, so they move quickly. This causes them to separate. Greater interaction with the stationary phase means a longer retention time. Greater interaction with the mobile phase gives a shorter retention time. This allows HPLC to separate and quantify. The solvent reservoir stores the mobile phase. It consists of water, buffers, or solvents. Each is stored in a clean, labeled glass bottle. Before use, solvents should always be filtered through a 0.45. Always remember, clean solvent equals stable baseline equals reliable analytical results. The next component is the degasser. Its job is to remove dissolved gases before reaching the pump. Why is this necessary? Because bubbles cause fluctuations and noisy chromatograms.
Now comes one of the most important components, the pump. The pump delivers the mobile phase through the system at a constant and precisely controlled flow rate. 1.5 milliliter min, a stable flow rate is essential. If it changes in times and peak areas shift, leading to inaccurate analytical results.
Now we introduce the sample. We use a syringe for manual injection. Load it and turn the valve. Auto-samplers do this now. Today, most pharmaceutical laboratories use auto-samplers. The analyst places sample vials into the tray. The instrument automatically injects an accurate volume. This follows the method.
This HPLC column measures 250 millimeters by 4.6. It is packed with 5 micrometer particles. Different compounds interact differently, causing them to separate.
The most commonly used detector is the UV visible detector. Other detectors include PDA, fluorescence, refractive index, and mass spectrometry. Once compounds leave the column, they enter the detector. The detector measures each compound as it passes through. It converts the signal into an electronic response. Here is our final chromatogram.
The detector sends data to the software. Each peak represents a separated braided compound. The retention time helps identify the compound.
Every successful HPLC analysis starts with the mobile phase. The mobile phase is prepared exactly according to the analytical method. Filter the mobile phase. Use a 0.45 or 0.22 micron membrane filter. Finally, degas the mobile phase. Use a vacuum degasser or an ultrasonic bath. To prepare a buffer, first, accurately weigh your chemical. Then, dissolve it in purified water. Finally, adjust the pH using an acid or base. After preparing the mobile phase, place the solvent bottles. Make sure they are in the correct channels. Check the tubing for bubbles or leaks. Now we are ready to begin. Let's turn on the HPLC modules. Power on the pump and autosampler. Now, column compartment and detector. Launch the chromatography software. Allow the system to stabilize. Set the column oven temperature, for example 30 or 40 degrees. A stable instrument provides stable analytical results. Let's prime and purge the pump. Before analysis, the pump must be primed. This removes trapped air. Purge each channel until no bubbles remain. After priming, confirm the correct flow rate. Check that the system pressure is stable and ensure there are no leaks. Never ignore pressure fluctuations. They may indicate air or a blocked filter. Today we discuss column equilibration. Next, install the correct HPLC column. Flush it until the baseline becomes stable. This is called column equilibration. Column equilibration allows the stationary phase to fully interact with the mobile phase. Skipping it may result in shifting retention times. Patience during equilibration saves time later. Now prepare the standard and sample solutions, exactly as described in the method. Always use calibrated balances, clean volumetric flasks, Class A glassware, and properly labeled containers. After preparation, filter the solutions, transfer them into vials, cap them securely. Good practices are essential. We first open the chromatography software. Then, we create the analysis sequence. Before analyzing test samples, perform system suitability testing. System suitability verifies that the HPLC system is performing correctly. Common SST parameters include retention time and resolution. They must meet criteria before sample analysis can begin. A chromatogram may look complicated at first, but it's actually very easy to understand. Let's examine its main parts. Vespinsion parts. First, we have the retention time. Next is the peak area, and finally, the peak height. Let's troubleshoot common HPLC problems, like high back pressure and peak tailing, as well as baseline noise, and finally, ghost peaks. Thank you for watching. Keep learning, keep growing, and remember, quality is never accidental. It is achieved through science, precision, and continuous improvement. See you in the next video.